Good morning. Welcome to the New England CEPAC webinar on oral and monoclonal antibody treatments for relapsing forms of multiple sclerosis. We're going to be beginning the webinar in 1 minute to give people time to join. Thank you. We'll be back in 1 minute to kick this off.
Hi, good morning. My name is Dr. Jason Waspy. I'm the chair of New England CEPAC. I'm a clinical cardiologist and health services researcher at the Massachusetts General Hospital. I direct our program in health outcomes, health services research in the MGH Heart Center. I'm also an associate professor at Harvard Medical School. I'm so happy to be here today. Multiple sclerosis is such an important issue for so many people in New England, throughout the country and the world. So this is incredibly important stuff that we have to deal with and discuss today. I'd like to begin by introducing other members of the New England CEPAC, and I would just ask as I go through the list, please introduce yourself and state, according to the sort of ICER policy on relationships with industry, please disclose any financial relationships with the industry. Each member of the panel has met our ICER conflict of interest policy. So we will just go through that as we go through the list. And as per prior in the Zoom generation, we will go through alphabetically. And I'd like to start with Dr. Rob Azeltine.
Good morning. I'm Rob Azeltine, professor and chair of the Department of Behavioral Sciences and Community Health at the University of Connecticut Health Center. I'm a medical sociologist whose primary research interests are in mental health and suicide, and I have no additional conflicts to disclose.
Thank you. Dr. Rena Conti. I think she may not be back, so let me just move ahead to Dr. George Goshua, new member of CEPAC.
Thank you. My name is Dr. George Goshua. I'm an assistant professor of medicine in the section of hematology at the Yale University School of Medicine. I run a lab that focuses on quantitative decision analytic modeling as well in the diagnostics and therapeutics in the hematology oncology space. It's a pleasure to be here today.
Thanks. We're really glad to have you join. Rebecca Kirsch.
Good morning. I'm Rebecca Kirsch from the National Patient Advocate Foundation, where I'm Executive Vice President of Policy and Programs, and I have no disclosures.
Dr. Steve Kogan.
Morning, everybody. I'm a professor at the College of Pharmacy and College of Business at the University of Rhode Island. I'm also the evaluation director for Advanced CTR, which is supported by an NIH infrastructure grant to accelerate clinical and translational research in Rhode Island. And I have no other conflicts or funding to report.
Thank you. Dr. Greg Lowe.
Good morning. I'm Greg Lowe. I'm a pharmacist and pharmacoepidemiologist, and I'm the director of the Mass General Physicians Organization's pharmacy program. And no updates to my disclosures. Thanks.
Dr. Aaron Mitchell.
Hello, I'm a medical oncologist and health services researcher at Memorial Sloan Kettering Cancer Center, where I treat prostate cancer and study determinants of care delivery in oncology and physician industry conflict of interest, and I have no conflicts of interest.
Thank you. Dr. Stephanie Nichols.
Good morning. I'm Stephanie Nichols. I'm an associate professor with the University of New England School of Pharmacy in Portland, Maine, an adjunct assistant clinical professor of psychiatry with the Tufts University School of Medicine, and core faculty for the Maine Medical Center Veterans Affairs Addiction Medicine Fellowship Program.
Dr. Jean Ryer.
Good morning, everybody. I'm Jean Ryer. I'm Director of the New Hampshire Citizens Health Initiative, and I lead the work on delivery system and payment reform at the University of New Hampshire Institute for Health Policy and Practice.
Thank you all for being
and
here.
No conflicts of interest.
Thank you. Thank you all for being here. And now we're going to move. We have a wonderful group of patient experts and clinical experts here today that I'm sure will enrich our conversation and deliberation. And I'd like to go through them and ask them to introduce themselves in order. And just please remember, after you introduce yourself, please declare any potential conflicts of interest. I'd like to start by introducing Dr. Bruce Cohen.
Good morning. My name is Bruce Cohen. I'm a neurologist specializing in multiple sclerosis and a professor at Northwestern University Medical School in Chicago, where I started a comprehensive MS program about 35 years ago. My interests are in clinical research and clinical trials with respect to potential conflicts. I'm a clinical investigator in the OPERA randomized controlled trial, which is now in an open label extension, which looked at ocrelizumab as treatment for MS, and which is considered in this document. I also have some common stock equity interests in Abbott Laboratory, AbbVie, and CVS Health. And thank you for having me here.
Oh, thank you for being here. Dr. Annetta Langer-Gould.
Hi, good morning, and thank you guys for inviting me to participate in this. So I'm the regional lead for translational neuroscience and practicing MS specialist at Southern California Kaiser Permanente. I was also a site PI for the ocrelizumab trial, but perhaps my most important disclosure is that I was the assistant medical director at Genentech, overseeing the rituximab and ocrelizumab development programs at the time when the company was still deciding which product to fully develop and pursue FDA approval for.
Thanks for being here. Next is Lauren Hirschfeld.
Hi, good morning. I'm Lauren Hirschfeld. I have been living with MS since I was 18 for about 9 years now. I also volunteer as a district activist leader with the National MS Society, and I am currently almost done with my PhD in medical neuroscience.
We're so glad to have you, and congrats on almost being done with the PhD.
Thank you, and thank you for inviting me, and I have no conflicts of interest.
No, it's our pleasure. Thank you for being here. It just enriches the conversation so much. Barry Talenti.
Hi, thank you for having me today. I'm Barry Talenti. I'm employed by the National Multiple Sclerosis Society as the Executive Vice President of Advocacy and Healthcare Access. And I also serve as president of the MS Coalition, which is a nonprofit coalition of 9 different organizations in the MS space. And I have no conflicts to disclose.
Wonderful. Thank you so much for joining us. Next, let me just introduce someone who needs no introduction, Dr. Steve Pearson, the president of ICER.
Thanks, Jason. Thanks again to our patient experts and clinical experts who will be joining us for today. So I'm going to give a brief overview and introduction to today's meeting. And we always like to start by reflecting again that the primary reason we're here today is based on people's lived experience with the condition. So here's just one quote. I've felt hostage to MS my whole life. People see that I'm well and don't understand what I'm actually going through. My treatments help, but they're an expensive hassle that I have to budget for every year. Treatment costs take the place of vacations, a new car, dining out. And as healthy as I may look, MS is a ball and chain. MS, again, like many chronic conditions, there's a huge variety and variation in the way that people experience it, that they can live with it, their family situations, how those are affected by it. But there's absolutely no doubt that MS creates a huge challenge for most patients, most people, and that it's the main reason we're living here is we're here, meeting today is because of the unmet need and the hope that we can bring new products, new options, new ways to help people. And that's the primary reason that we start. On the other hand, we're also here today for other reasons, because what happens the day that these treatments like ublituximab, which was approved by the FDA just a couple of weeks ago, when that happens that day, there is a completely welcomed sense of celebration. It's a celebration of having something new to potentially help people who haven't been able to have successful treatment before. It also represents the success in some ways of the commitments of drug makers, of investors, of scientists, research scientists, and patients and their families who've participated in the evidence development over many years and taken risks and sacrificed, et cetera. So there's a real sense of celebration. At the same time, that day that the FDA approves a new drug, there are still important questions on the table because it's new. And so there's still, residual questions around, do we know, what do we know about the risks and benefits for specific types of individuals? And since the FDA frequently focuses on the drug to a certain extent in a pretty narrow framework, other questions exist around how these new treatments will fit into the evolving landscape of other available treatments. Also, and as you heard in part from the patient, from the person living with MS, from their testimony, for all conditions that are chronic with drugs that cost this much, there are questions around reasonable prices and costs to patients themselves, to the health system, and to the government, which often ends up paying a large proportion. So all these questions are still on the table, and we need to look for lessons that we can learn from this new entrant and in context with the others to not only make decisions today, but to make sure that we try to talk about the structures, the programs, the policy changes that we might want to make sure that we do this even better in the future. So all this is a lot to talk about, but there's another reason we're here today, which is, in addition to thinking about the cost for individual patients, there is an impact of rising healthcare costs across the board for everybody, no matter what healthcare system they're part of. And I know I presented some of these patients' stories before. They're in the public domain, and they came out with an article kind of summarizing a report last year from Kaiser Health, and from the Kaiser Family Foundation. And what this survey did, they did a national survey, and they don't survey all Americans, they do a representative sample. And by the findings, they could estimate that more than 100 million Americans today live with medical debt. And by that means that they were asked, do you have medical charges that you are not able to pay off today? And so that incorporates a lot of people. It incorporates people who are bankrupt because of medical costs. It incorporates people who are carrying charges on their credit card and not able to pay them off. A lot of people. And these people are almost never in the room when we're talking about, honestly, when we're talking about coverage, even when we're talking about pricing or payment, et cetera. They're unfortunately kind of a hidden problem too often in our society, but they are there. They're in your community down the block, et cetera, if it's not you yourself. And so, these stories, Marcus and Allison Ward on the left with their kids, they ran into trouble with premature delivery, and they had expenses, both drugs, but also other expenses, and felt that they needed special care. And anyway, they ran out of the ability of their insurer to cover that and had to move to live with family across the country, and they are currently in significant medical debt. The couple in the middle, Sam and Ariane Buck from Arizona, his job is to call people to enroll them in Medicare Advantage programs. And he was in between jobs when he got sick and had to go to the ER and ended up getting into medical debt. And afterwards, they had 2 cases in which both of them were turned away from a doctor's office because of their medical debt. And this survey actually found that 1 in 7 of the people out of this more than 100 million people said that they had either been denied care at a hospital or at a doctor's office because of their medical debt. And 2/3 of that 100 million Americans said that they had themselves postponed or given up care prescribed by a clinician because of their medical debt. So this is a serious problem. Sherry Foy, the woman on the far right, had to declare medical bankruptcy with over $600,000 in medical debt. She was on an employer program with a cap on lifetime costs. So yes, these are problems with the insurance system, but it's just a statement that every time we kind of talk about what we're doing with innovation and how we want to do the right thing for patients, we have to view this in a broad context that includes all of us. So today, we're going to talk a lot about MS. We're going to talk about families and individual people. And yet we have to remember that around them, around all of us, are other families with other medical issues, life issues, and we have to figure out a way to welcome innovation and do the best thing for the person in the center of our attention, while keeping a balance across the system that affords and allows future innovation for others. So we're here today to wrestle with those issues and a lot of different perspectives. You've already been introduced to the members of the New England CEPAC. Just a few words about ICER, which is the kind of academic and hosting organization. Our funding, which we update twice a year, has just been updated again. So this new pie chart, which we keep on our website, shows that of our overall revenue, 65% now comes from nonprofit foundations. The primary one within that is the Laura and John Arnold Foundation, but we also have grants from the California Healthcare Foundation, from the Commonwealth Foundation, and some others. The next kind of largest piece of the pie, if you will, you can combine the 10% from health plans and provider groups with 15% from life science companies. We use that for a separate policy summit and evidence webinar program for them. So that does not go into the support for the research that we do on new drugs. We're also about 8% now of our revenue from subscribers to our cloud-based platform of all of our work called ICER Analytics, and a small sliver from individual philanthropy. So the report today, that's the foundation in some ways for the discussion, has taken almost 8 months to get us from the beginning to now. And it starts with a very intensive couple months phase of scoping, during which we really try to ask and listen. Even if we've done a topic before, we want to make sure we're listening to what's new, but we seek guidance from patients and patient groups on the lived experience with the condition and what matters most to patients. We talk to clinical experts, obviously, we talk to the manufacturers and other stakeholders. Then once we kind of have our orientation from that process, we start the evidence review, looking at all kinds of evidence, whether it's been published or not. We try to be omnivorous in our attempt to try to get the right kinds of evidence into the thinking around value. And we do create a cost effectiveness model that will try to take whatever we have in the short term and spin that out into the future to try to make sure that we capture the longer term benefits and cost effects of new treatments. This report goes through several iterations of public comment and revision. And then we were very fortunate to have a rich group of expert reviewers that you can see here, including the clinical experts on the call today, but other folks, I'm not going to read every single name, you can see them there, but it's intended to get a broad perspective of experts on our report before it's finalized. Now, the report structure that is intended in some ways to support today's discussion can be pictured around our value assessment framework, because ultimately, we'll talk about clinical effectiveness, cost effectiveness, all these other pieces are elements in thinking about value and how we recognize, reward, and pay for value in the right ways. So the report is built upon different building blocks around, in a sense, what new treatments can do for us. And at the bottom, they start with new treatments can help us live a longer life. They can help us, with health benefits. They can, even if they don't do that, or if they do, they might also help us return to higher function levels. They might have fewer side effects than other treatment options, so they might have a variety of different benefits. When we look at cost as part of value, we are not just thinking about the cost of the drug, because it's important to think about costs across the whole healthcare system, for drugs in particular that might help prevent future hospitalizations or reduce doctor office visits. We need to look at all of that broad effect, and we often try to do the best we can to also even think about costs that spill over outside the healthcare system, the effects of treatment on people's ability to work better or to attend to school, those kinds of things. The blue boxes there represent elements that for which sometimes there's evidence, but sometimes there's not, but they're still important perspectives or parts of value. One that we call benefits beyond health, or in a sense, things that in some ways they are health, but they might be missed. They're never going to be captured by the usual clinical trial that's done for FDA approval. These are things like the longer term effects on people's ability to pursue a long-term kind of job approach or an educational program, caregiver effects, those kinds of things. And then at the top, we list the idea of special social or ethical priorities. These can be framing issues around the relative severity of a condition and how much priority we want to place on a condition because of its history of discrimination against patients, or because, again, of its severity, the short-term risk of death, other reasons that we might or might not want to give specific priorities to certain kinds of treatments. So all of this is discussed and explored in the report, but it'll be a rich part of our conversation today. We're going to start off with presentation of the clinical evidence by Dr. Grace Lin, followed by the presentation of the cost effectiveness, the economic model by Dr. Melanie Whittington. Then public comments and discussion will be followed by lunch at noon Eastern. And then we'll return for further deliberation on this evidence by the CEPAC before they take votes on the clinical effectiveness and value. Following a short break then at 2 o'clock Eastern, we'll start a policy roundtable. And that's where the patient experts, clinical experts, will be joined by payer representatives and the manufacturer of ublituximab in this case, to really talk what it looks like going forward, to put the evidence into practice through coverage, through education, through future research decisions in a way that can get us to that best point of maximizing our ability to achieve fair pricing, fair access, and future innovation in this space and others. So we'll do that and then be concluded by 4 o'clock Eastern time. With that, let me hand it off to Dr. Grace Lin, who will, again, as I mentioned, begin with the presentation of the clinical evidence. Grace?
Unmute myself. Sorry. Thank you, Steve. Good morning, everyone. As Steve said, I'm Grace Lin. I'm the Medical Director for Health Technology Assessment at ICER, and I'm also a Professor of Medicine and Health Policy from the University of California, San Francisco. And I'll be taking you through a brief summary of the clinical evidence. Next slide, please. This report wouldn't have been possible without my collaborators, Dimitri Nidikin, Serena Heron-Smith, Avery McKenna, and Felucio Agbula. And I have no disclosures other than I received funding from ICER for this report. Next slide, please. So as Steve said, we're here to talk about multiple sclerosis, which is an autoimmune disorder of the central nervous system affecting almost 1 million Americans. It is often diagnosed in young adulthood in people in their 20s and 30s and has a female predominance of about 3 to 1. Because it's often diagnosed during young adulthood and people live quite a long time with MS, the total economic burden of MS in the US is quite high. Annually, it's about $85 billion with direct medical costs accounting for the bulk of that economic burden. MS is a demyelinating disease, and since you have neurons, and since you have neurons throughout your body, multiple organ systems are affected. For example, persons with MS can experience numbness and pain, as well as muscle spasms, which cause difficulty with balance and walking. We heard from persons with MS that fatigue and cognitive function are also often prominent symptoms, and also that bowel and bladder symptoms can substantially affect quality of life. Next slide, please. Symptoms of MS impact physical and emotional health and work, education. And again, since this is a disease that manifests in young persons, it affects family planning as well and social and leisure activities. Many of the symptoms aren't immediately visible, as I mentioned, the cognitive symptoms, for example, and they're a challenge to manage in daily life. The economic impact includes lost wages from missed work. Some people need to transition to part-time work because of their symptoms, or leave the workforce early. And then there can often be high out-of-pocket costs for medication and assistive equipment. The main goals of treatment are to preserve function and remain as independent as possible. Since some of the treatments are not treated by what we call the disease-modifying therapies, which is what we'll talk about today, and they may require other medications, persons with MS may end up taking actually quite a bit of medication, both disease-modifying and symptomatic medications. Treatment options and shared decision are important because there are a lot of treatments in this space. And we heard from persons with MS that some of them have a strong preference for certain types of medications like oral agents or injectables. Others prefer infusions. So there's a lot of choices here. And then there is heterogeneity in the disease symptoms and disease course. And so therapy is often, it should be based on individual disease history and personal characteristics. Some of the main measures of disease activity that we'll talk about today focus on the ability of a patient to ambulate. But persons with MS who've lost that ability to ambulate get concerned that they will not receive more aggressive treatment, because the thought is that now they are not able to ambulate, so their disease has progressed and they may not benefit from these treatments. One person with MS told us, "I am relevant, I just don't want to be forgotten when I'm at that stage." So that is one thing that we heard from persons with MS for in terms of treatment choice. Next slide, please. Standard of care and management include both disease-modifying therapies, as I said, and supportive care such as symptomatic medications, psychological support, management of comorbidities, and lifestyle interventions and rehabilitation. There is some uncertainty surrounding the ideal therapeutic approach for disease-modifying therapies. So this is actually currently being studied in randomized clinical trials of whether you should do what's called escalation therapy, where you start with a DMT that's of more moderate efficacy with maybe a more tolerable safety profile, and only go to a more aggressive therapy if the disease starts to progress. Or there's what's called induction therapy, where you start with a high efficacy DMT with higher risk of serious adverse effects at the beginning, and then either continue that or consider stepping down to a less aggressive therapy when the disease is controlled. Next slide, please. So the scope of our review today, we compared the clinical effectiveness and safety of oral and monoclonal antibody DMTs for the treatment of adult patients with relapsing forms of MS. And relapsing forms of MS includes RRMS, which is a periodic relapses with complete or near recovery, and SPMS, which is progressive worsening that people sometimes transition to following RRMS. We are focused on first-line therapies and new clinical evidence since ICER's 2017 MS class review, but while we've included comparisons in this report, and we'll mention them briefly here, this presentation will be centered around the newest monoclonal antibody, ublituximab, which was just approved by the FDA a few weeks ago. We focused on ublituximab because it has not been compared to the other more established treatments, and because it was, as I said, just recently approved. Next slide, please. So you'll see in this slide that we're going to discuss ublituximab as the main intervention in the clinical effective section. Ublituximab belongs to the anti-CD20 class of monoclonal antibodies and is infused once every 6 months after an induction period during the first year. We will compare ublituximab to other oral and monoclonal antibody DMTs. In the red circle here, these are the oral medications, the sphingosine 1-phosphate receptor modulators, that's a mouthful, so I'll say S1P receptor modulators, the fumarates and teriflunomide. Next slide, please. And then the monoclonal antibodies, which include the anti-CD20 drugs, ocrelizumab, ofatumumab, and rituximab, and natalizumab. Next slide, please. In terms of clinical outcomes, we are going to look at the annualized relapse rate, which is the per person average number of relapses in 1 year. We'll also look at confirmed disability progression at 3 months and 6 months, which is we'll call CDP either 3 or 6. And this is characterized by the worsening of neurologic deficits measured by changes in the expanded disability status scale, the EDSS, which is a 10-point scale that assesses for disability in persons with MS, with higher scores indicating greater disability. This is based on assessment by a neurologist, and it is the standard, one of the standard outcomes in clinical trials. We also looked at MRI outcomes, including the number of total and new T1 and T2 lesions. And we looked for also patient-centered outcomes such as confirmed disability improvement. There's a scale called the MS Functional Composite and a scale called MS Quality of Life, which are MS-specific quality of life scales. And then the SF-36, which is a generic quality of life scale. Next slide, please. So let's dive into the clinical evidence. So first, I'm going to talk about ublituximab, some direct evidence we have from the phase 3 randomized clinical trial of ublituximab called Ultimate 1 and 2. These were double-blinded controlled trials of more than 1,000 patients comparing ublituximab and teriflunomide, which you'll recall is one of our oral medications. And this is done in patients with relapsing forms of MS. As you can see in the table here, we've looked at the outcomes of annualized relapse rate, or ARR, and ublituximab had significantly lower ARR than those treated with teriflunomide. For the outcome of CDP6, we have a hazard ratio here of 0.66. Patients, meaning that patients treated with ublituximab had about a 34% less chance of progression at 6 months compared to those treated with teriflunomide. Although, as you can see from the 95% confidence interval, this result was not statistically significant. The CD3 data, which I haven't presented here, are similar. The investigators also assessed whether participants were free of disease activity, or this NADA, which is no evidence of disease activity. This is defined as no relapses, no MRI activity, and no worsening of disability between weeks 24 and 96. And people treated with ublituximab had 5 to 8 times, were 5 to 8 times more likely to have no evidence of disease activity compared with the teriflunomide group. Next slide, please. In terms of MRI outcomes, the mean number of lesions for T1 and T2 were lower in the ublituximab group versus teriflunomide. And quality of life was measured, the MS quality of life and the SF-36 were measured at week 96, and those also favored the ublituximab group in all subcomponents compared with teriflunomide. Next slide, please. So we have that direct evidence of ublituximab compared with teriflunomide, but we were interested in how ublituximab might look compared with the other DMTs, because as you've seen, there are a lot of DMTs for MS and not a lot of head-to-head comparison trials. So in order to compare the agents to each other, we used methods for network meta-analysis for indirect comparison. So during our systematic review, we found sufficient data to run an NMA model for the ARR and CDP outcomes using the Bayesian Markov chain Monte Carlo method. And our base case used random effects, and we did some sensitivity analysis using other methods. Next slide, please. In terms of key baseline characteristics of our trials, there were a lot of trials, so I won't, you can look at the report if you want all the details on each trial, but the trials ranged in size from relatively small trials to very large trials, and they ranged in duration from 48 to 120 weeks. The population was primarily younger, female, and white populations with RRMS. And people with active disease who had a baseline relapse rate in the past 12 months of 1 to 1.5 relapses and a baseline EDSS score of 1.6 to 3. Participants in the trials also were mostly treatment naive, meaning that they had not been treated prior with a DMT. A couple of the notes on the NMA. 3 drugs were excluded from our NMA from the beginning: monomethyl fumarate and diroximel fumarate, which are part of the fumarate class. They were excluded because their FDA approvals were based on showing equivalent effectiveness to dimethyl fumarate. So throughout this presentation and the economic presentation, you'll notice that we use dimethyl fumarate as the example of the fumarate class. Also, cipatamod was not included in the NMA because its pivotal randomized controlled trial was done mainly in SPMS patients, so its population was different from the other drugs in the NMA. Next slide, please. So here we have the NMA results for the outcome of ARR. Here we include 19 phase 3 trials and 1 phase 2 trial that was of rituximab. We calculated the rate ratios comparing ublituximab compared to all the other DMTs. This red box here shows you ublituximab and the other monoclonal antibodies. And as you can see, the dots are all fairly close together and their confidence intervals cross 1, and they're all fairly close to 1, meaning that there was no statistically significant difference in reducing for ublituximab reducing ARR compared with the other monoclonal antibodies. Next. This next group of drugs are the oral drugs. And when we look at the effect of ublituximab compared to the oral medications, such as the S1P receptor modulators, dimethyl fumarate and teriflunomide, we see that in general, ublituximab was around 30 to 50% better than the oral medications for reducing ARR. And the other thing that we can see from this forest plot is that the oral medications are somewhat less effective in reducing relapse rates than the monoclonal antibodies, but they're better than placebo. Next slide, please. Here we have the results for 6-month CDP. And again, this top box here are the monoclonal antibodies. You'll notice here that rituximab is not included. So in this network meta-analysis, we included 18 phase 3 trials. We did not have reliable CDP data for rituximab, so we did exclude rituximab, so you won't see that in the results. At 6 months, you could see that ublituximab was no better than ocrelizumab, natalizumab, and ofatumumab. Numerically, perhaps a bit worse than natalizumab, ocrelizumab, but as you can see, those confidence intervals are very wide. Next slide, please. Compared to oral agents, ublituximab was, again, numerically maybe slightly better than oral agents, but none of these differences was statistically significant. As you can see, all those confidence intervals are wide and cross 1. Next slide, please. In terms of MRI outcomes, we found that MRI outcomes weren't measured consistently enough across trials to run an NMA comparing the different disease-modifying therapies, but qualitatively, all the DMTs either reduced the total number of new lesions or on MRI, or both the total number and the number of new lesions. And the monoclonal antibodies in general, again, appeared to be slightly more effective than oral medications on MRI outcomes. Next slide, please. One of the common critiques of MS trials is that the main outcomes of ARR and CDP don't fully capture the patient experience, including cognitive function, fatigue, mobility. Again, we found that these weren't measured consistently across clinical trials, so we couldn't do any quantitative comparison. We did do some qualitative summarizing in the report, and you can find details of that in our report. I'll just mention here a couple of highlights that in terms of ublituximab was and fingolimod actually both used the MS functional composite, and there was a difference, statistically significant improvement for ublituximab and fingolimod in the MS functional composite over their comparators. confirmed disability improvement, or CDI, is an emerging trial outcome. And this allows for the fact that people with MS might actually, their function might improve or on DMT. And this was explored in the ublituximab trial, and there was a greater number of ublituximab patients that showed improvement in disability versus teriflunomide. Although again, this was an exploratory outcome. Next slide, please. In terms of harms, for ublituximab, infusion reactions and respiratory tract infections were the most common in the RCTs. And there's a slightly greater incidence of serious adverse events and discontinuation due to adverse events compared with teriflunomide. Because treatment with ublituximab can reactivate hepatitis B infection, hepatitis B virus screening is required before starting ublituximab. Next slide, please. Here are the harms, just a summary of the harms. More specific harms can be found in our report. But for the monoclonal antibody class, there's an increased risk of infection due to B cell depletion. There are commonly infusion and injection related reactions. And then in terms of black box warnings, for natalizumab, there's a black box warning for a condition called progressive multifocal leukoencephalopathy, or PML. And then that is also seen with rituximab on the rituximab label, although because rituximab is not FDA approved for MS, it's unclear whether that also applies to MS patients. For the oral therapies, we can see that the fumarates can cause flushing and gastrointestinal events. The S1P receptor modulators can cause cardiac events, so that you need to monitor the person when taking the first dose. And teriflunomide has a couple of black box warnings for liver toxicity and toxicity to embryos in pregnant women. Next slide, please. In terms of controversies and uncertainties for this report, so there was a lack of long-term efficacy and safety data for ublituximab compared with some of its comparators. For example, natalizumab was approved in 2004, so there's a couple decades of data. Rituximab, as I mentioned, is not approved for MS, so use of it is off-label, and there is we found there was a new phase 3 trial for rituximab that we included in this report to augment our understanding of the effectiveness of rituximab. There's a lack of head-to-head data amongst and between classes, and trial heterogeneity may affect the NMA results, so that but we did do some sensitivity analysis that showed no evidence for effect modifications, and our results were consistent with other published network meta-analyses. And then finally, patient important outcomes were really not measured well across trials. Next slide, please. As always, we look at potential other benefits and contextual considerations. MS is a chronic disease, and so its impact is large over the lifespan. And we've already, mentioned the impact on education, work, and family planning. The caregiver impact gets progressively large as a person loses mobility. The treatment burden is large, particularly both in terms of daily life, day-to-day life, and length of treatment. Black Americans with MS may experience poor outcomes. It's not entirely clear whether this is due to different disease characteristics or poor access to care. And then COVID-19 had a pretty significant impact on persons with MS. There were delays in receiving care, particularly infusions, when the healthcare system was sort of overwhelmed and canceling anything elective. And then the B cell depleting therapies, which are the anti-CD20 monoclonal antibodies, have an impact on a person's response to vaccines. Next slide, please. So we did receive some public comments, and as always, we appreciate the comments we received on the draft evidence report. Here's some of the major comments. We got the comment that other trials should have been considered for inclusion into the NMA network. We did, as I said, run sensitivity analyses and showed that including and excluding trials did not substantially change our conclusions. We also received the comment that rituximab should not be included in the review due to the lack of FDA label for MS. Rituximab, however, is frequently used off-label to treat MS, and we did find new randomized controlled data, so we thought it was worthwhile including in this review. In terms of longer-term evidence and other patient outcomes, we received comments to consider those, and we looked hard for other patient-relevant outcomes for evidence. And then in terms of real-world evidence, we focused on the randomized trial evidence here, but we did include real-world evidence such as for harms were available. Next slide, please. In summary, all the DMTs are effective in reducing relapses. Ublituximab appears similar to other monoclonal antibodies and better than orals and placebo. The impact on disease progression, or CDP, is less certain, but ublituximab and the monoclonal antibodies may be slightly more effective than the oral DMTs. The harms differ across drugs and classes, and ongoing trials are going to help us answer questions about treatment sequence and what then the appropriate point of discontinuation. Next slide, please. So this leads us to our evidence summary, our evidence ratings, excuse me. And again, we're talking about ublituximab versus all the other drugs. So based on a review and the data I've presented, we judge that there's insufficient evidence to distinguish ublituximab from the other monoclonal antibodies. And we feel that the evidence demonstrated that ublituximab was comparable or better, the C++, than the fumarates, fingolimod, ozanimod, ponesimod. Those were the oral medications in the NMA that we included in the NMA. Again, we had to exclude siponimod because of its population difference. And so we have insufficient evidence, or I, for that comparison. We had direct evidence of ublituximab compared with teriflunomide from the randomized clinical trials where ublituximab had improved outcomes versus teriflunomide. So we gave that rating a B, that it had represents incremental improvement over teriflunomide. And then finally, because MS is a devastating disease, compared with a placebo or no DMT, we judged that ublituximab provides superior improvement, leading to an A rating. So that concludes my brief summary of the clinical effectiveness data from our review, and I'm happy to take questions. Thank you.
Thank you, Dr. Lin, for the presentation. I just first wanted to interject. Dr. Rina Conti's camera was not functioning well at the beginning, so I just would like her to introduce herself. She's a CEPAC member.
Thank you so much for the opportunity to be here and to introduce myself. Apologies for being late. My name is Rina Conti. I'm a health economist and associate professor of markets, public policy, and law at Questrom School of Business at Boston University. I also run a technology policy center here at the university between economics faculty and law school. I'm delighted to be here today. Thank you.
Thank you. And with that, we have about 12 minutes for questions and discussions before the economic modeling presentation. So just wanted to see if any of the clinical experts that were listening or the patient experts had anything they wanted to add before we go to questions from CEPAC. Okay, hearing none, maybe I'll just start off by asking, for either for Dr. Lin or again the clinical experts, or Lauren, who's both a scientist and a patient. I have a question about clinical endpoints. So obviously in comparative effectiveness research, the best endpoints to look at are hard endpoints, things like mortality or symptoms that matter to patients. That's always ideal. But then sometimes you have this problem where you might have a conservative bias in a comparative effectiveness analysis, which is to say that some sort of surrogate endpoint might actually be meaningful and might lead to changes and differences in harder outcomes over the long run, but you're not seeing it yet. So I will say, just by looking at these data, I have a little bit of that vibe from the numbers. So like the hazard ratios for the MRI lesions seemed unbelievably different, which is to say that I guess I'd like to know from all these experts, and from Dr. Lin, what endpoints are most meaningful and important? And could we be making a mistake in the evidence rating to be too cautious because we're focusing on the real clinical endpoints as opposed to MRI lesions? It is an inflammatory disorder. One would imagine that less inflammation leads to less symptoms. But I know that surrogate endpoints don't always translate into hard endpoints.
Thanks for that question, Dr. Bossi. Let me just start by saying that yes, this is MS is complex, and there's clinical outcomes and there's the MRI outcomes. We heard from patients that the MRI outcome, the MRI lesions don't always correlate with all their symptoms. And so that they were concerned that judging whether somebody had quote unquote active disease based just on MRI outcomes would be somewhat misleading, because some of those symptoms don't correlate well with that. And so the other issue here is that the clinical trials are set up based on FDA guidance, and the FDA guidance is that annualized relapse rate and confirmed disability progression based on EDSS are kind of the standards, and so all the clinical trials are set up that way. And so there is this disconnect between whether or not those actually capture the entire patient experience. I see that Dr. Cohen can further comment on that.
Yes, thank you for the question, because I think it's very important. The endpoint, the frequency of endpoints obviously impacts the ability to discriminate between the different interventions, and the MRI endpoints are much more frequent than the clinical endpoints, so that helps give you a better response in terms of discriminating between the agents and potentially licensing the drug. Relapses are also relatively easy to measure. Disability, on the other hand, has limitations because the EDSS is a categorical scale, and it is defined by certain changes which actually represent only the threshold with which the underlying biology of the progression of MS emerges to the point where we can actually measure clinical differences. And since clinical differences are necessary in order to get an indication under FDA guidelines, that's why the EDSS becomes the primary outcome measure for clinical disability. But in fact, we know from analyses that have been done of clinical trial populations that much of the impact on disability occurs not at the time of the relapses, but during periods of relative quiescence during the clinical trial. Nonetheless, we also know that relapses clearly affect the risk of disability. So what this means is that suppressing relapses is important, but it appears that the suppression of relapses in terms of the disability outcomes is most prominent early in the course of the disease. And although it continues to be relevant later on, the best effect on disability occurs early. What this means is the trajectory of disability over time may be different than what we see in the 2 years of a randomized clinical trial. In fact, those trajectories may separate at an increasing slope as time goes on, depending on what the initial therapy is. Thank you.
So thank
This
you.
is Annette. This is Annette. I think the MRIs, the reason those are really important clinical secondary outcome measures is they're objective. So relapse assessments and disability assessments in the context of clinical trials and certainly clinical practice, these are not, they blind, but they're not truly objective measures. They can easily be influenced by treating physician and examining physician bias. And in the case of EDSS, there's a lot of inter-rater reliability problems, 1 point, in the lower end of the EDSS scale, which is what we're talking about in relapsing remitting MS trials. These trials are typically recruited primarily out of Eastern Europe. Often they are not even neurologists doing the exam. And the examining physician may change multiple times during the course of the clinical trial, which is typically on order of 2 to 3 years in duration. And so we think of if we saw only an effect on relapse, but no effect on MRI, or only effect on disability and no effect on MRI, then we'd start to question whether there were some irregularities in in the data collection in the trial. So we like to see that MRI effect as a sign of consistency of treatment effect. And there's a lot of variability based on entry criteria and the population recruited. So typically in the older trials, they have much higher MRI disease activity. Now that most patients in Western countries in the United States are with highly active disease are on therapy, those patients are harder to find.
I'd like to echo that having that MRI endpoint, while yes, is very objective, I, in my personal experience, have not shown more disease on MRI in many years, but that doesn't mean my chronic pain or my fatigue haven't worsened, because they have. So I would like to echo that these kind of secondary measures or secondary endpoints often don't follow MRIs and often aren't harmonious with that.
Those responses are so helpful, so we appreciate the context very much. Are there other questions from the CEPAC? Please use the raised hand function. Or I think I can see if someone just waves, either way. Dr. Conti.
Thank you. Are there real world evidence assessments of disease progression on each of these therapies that are being used as comparator? And how can you, and if so, can you tell us a little bit about what that evidence suggests in terms of endpoints that matter to patients and to also providers?
So this is Annetta. So we just completed a large prospective cohort study in the country of Sweden, comparative effectiveness of rituximab versus all other commonly used DMTs. So that included fingolimod, dimethyl fumarate, natalizumab, and the traditional injectable therapies. And at the end of 3 years, I think the most striking, so in a real world population, so this is relapsing remitting MS patients, either we divided it into 2 groups, either they're starting their very first therapy or they're switching for the very first time. And I think the striking findings were that at the end of 3 years, disability progression rates are much lower in real world populations in this 100% treated patient population than they are in the clinical trials population. Patients are also somewhat younger, they have shorter disease duration. It's an idealized healthcare system. Everybody has health insurance. Everyone is seen by an MS specialist. This does not happen in the United States, and everyone's getting an annual MRI scan. First sign of trouble at relapse or MRI disease activity, they're escalating these patients primarily to rituximab, but also some of them to natalizumab therapy. At the end of 3 years, less than 50% of patients were still on dimethyl fumarate. The highest staying power and adherence was still with rituximab, and about 75% of patients were still on natalizumab. I think that's really important for American populations. If you start someone on dimethyl fumarate, you can expect that more than 50% will no longer be on it in 3 years, and that may happen when you're not looking. And they may end up on nothing during that time and then have uncontrolled disease activity. In terms of outcomes that matter to patients, treatment satisfaction surveys treatment satisfaction was improved in the rituximab treatment group, not terribly surprising over other ones. And then we're still analyzing the fatigue outcomes. But in general, you know, these treatments, as Lauren pointed out, they're not aimed to treat symptoms. So they're not aimed to treat pain or fatigue. The best thing we can do is prevent them from having another relapse that would result in neuropathic pain. But in patients who already have it, we don't really expect improvement in that based on the anti-inflammatory treatment that they choose.
I'd like to add to that, because I agree with Annetta's comments, and would like to add that there's a very large published real world observational study coming from the MS Bates Coalition, which is a Western European coalition in which prospectively acquired data is obtained in MS centers for patients and pooled. And this looks at initiation of high efficacy therapy versus moderate efficacy therapy, and finds that using the EDSS, and these are raters who are certified to do the EDSS to try to minimize bias that's inherent in the scale. It shows benefit on disability outcomes as measured by EDSS up to 10 years later. That data is published.
Yeah, there's some small studies. I'm just going to say MS based, 50% of the data is missing, and it's a passively enrolled observational study. So they're not actually protocolized studies the way that the comparative effectiveness study that we did with PCORI funding in Sweden is. But the results are all very similar, that starting on a high efficacy therapy, B-cell depleting therapy or natalizumab at onset, the patients have less relapses, less MRI disease activity, and less disability accumulation in intermediate outcomes at 3 years. But nobody has 10 or 20-year data.
Thank you all very much. Jean Reier.
So could you dig a little bit more into comparative harms, and especially from the patient experience point of view?
I mean, I think there's no question that the more effective the medications are at suppressing disease activity, the higher the risk of infections is. In terms of sort of treatment-specific infectious risk, it's really PML with natalizumab, which is a often fatal, always permanently disabling disease. And then with fingolimod, oddly enough, fingolimod and S1P1 inhibitors, it's herpes family viruses. So there were a couple deaths with varicella zoster, and that has been shown repeatedly both in the clinical trials and in the observational settings. So we typically won't put somebody with recurrent herpetic, recurrent cold sores or genital herpes outbreaks, frequent recurrent outbreaks onto an S1P1 inhibitor. And patients with rare outbreaks, we would put them on prophylactic antivirals. So the interferons and glatiramer acetate are still by far and away the safest medications for patients. And there are still patients who do very well on these therapies. The concern really is just on a population level, they don't do well enough. So if you're in my situation, you have to take care of, you're trying to organize a system that takes care of 12,000 MS patients, only under certain circumstances would we recommend starting with one of those medications. And in most of them, we're going to recommend starting with rituximab and then looking for the induction and then looking for the lowest effective dose.
With respect to the PML risk applies to virtually all of the medications that are listed. There are actually more cases of PML in patients taking fingolimod and dimethyl fumarate than there are in patients taking ocrelizumab, if you exclude cases that were related to prior treatment with either fingolimod or natalizumab, just switching to ocrelizumab. There are only 2 cases with ocrelizumab that are unrelated to other drugs. There are 7 cases with dimethyl fumarate at the last, to my knowledge, and there are over 30 cases with fingolimod. So from the standpoint of serious opportunistic infections, there does not appear to be a high increased risk to date with our experience with B-cell depleting therapies in MS patients.
Thank you. I think Lauren has a comment and then George has a question, and then we'll move on to the economic presentation. So if I can just start with Lauren, please.
Thanks. Yeah, I just wanted to speak on the patient perspective of that kind of risk benefit decision. For transparency, I am on dimethyl fumarate. I have been for almost 9 years with a very brief stint on diroximel fumarate. And a lot of the decision to take those medications really came, like you said, from the PML risk, which is lower for, say, these fumarates than for Tysabri, for natalizumab. PML, when you're faced with an already degenerative, disabling disease, and then being told that a side effect might speed that up is very scary. There's also the issues with S1P. When I was being offered fingolimod, I was told I'd have to have my heart monitored for the first dose because it could induce a heart block, which is also frightening. And then even with acrelizumab, some of my peers who are on those infusion drugs have told me about the increased cancer risks, which again, you have this degenerative disease and then you're faced with something just as bad. So there is that kind of risk benefit. How much will this hurt me, but how much will it help me?
Thank you. And this will finish up this part of the session with a question for Dr. Goshua, and then I'm going to start the economic section.
Thank you very much. I just wanted to ask our clinical experts the heterogeneity that's present in our country, in the United States, because what I'm hearing about real world data that informs real world evidence, a lot of it seems to come either from Eastern Europe, Western Europe, or Northern Europe. So when you're counseling your patients, do you feel like the generalizability of that data does apply to the United States, given our much more mixed and heterogeneous patient population?
So I think the Western European data does. I think data such as Annetta described does, because if you look at the populations that are most likely to have relapsing MS, they are in fact people with European ancestry. So I think that, you know, from a genetic perspective, I think that data probably does apply.
So I'm going to disagree with that. We've actually published first in 2013 and then most recently that the incidence and prevalence of multiple sclerosis is similar in young Black and Hispanic patients as whites. Maybe it's actually higher in African-American women, and that over time, the prevalence of multiple over age groups, the prevalence of multiple sclerosis is similarly high in Black Americans as it is in white Americans. Black Americans are far less likely to have health insurance. Young patients have a lot of difficulty sometimes accepting the diagnosis, and we have a real issue. So I'll just say that I should back up and say some of that safety data, it does come from Southern California Kaiser, and our patients are sicker. Our patients have more comorbidities, and the risk of infections, at least the way we count it, which is slightly different than the Swedes, is higher in the American population of MS patients. The overall sort of comparison with other treatments looks very similar. So I do think our patients, they have more comorbidities that put them already at higher risk of multiple sclerosis. We also use it in more disabled patients than they do in Europe. And that's where we see the highest risk of serious infections. I think the biggest issue is around staying power and cost, because we have a very fractured healthcare system where many of our African-American and Hispanic and otherwise indigent patients have they have inconsistent access to neurological care and to treatments. And drugs like dimethyl fumarate are not realistic solutions because they need to have continuous health insurance to pick it up, even if it's cheap. And they have to be able to take it twice a day. And that, for many of them, it has a lot of side effects. It's also not as effective. They're more likely to have breakthrough disease. For many of them, it just ends up being an unrealistic solution. So even though it's cheaper than biosimilar rituximabs, we found that biosimilar rituximabs really are the answer for addressing disparities in outcomes in Black and Hispanic Americans.
Thank you. We are a few minutes over, but Barry, I see your hand up. Do you want to, if you have a brief comment, then I'll move on to the economic presentation.
Yeah, I would just echo many of Annetta's comments. I think it's clear that we have a lack of diversity in clinical trials. And so we don't have all of the answers that we need for Black and Hispanic Latinx people living with MS. We just don't know. We're making generalizations based off of a largely white population in clinical trials. And then as Annetta shared, the healthcare system in the United States means that people are often sicker, they have comorbidities, we have much higher discontinuation rates and lack of adherence than we would see in other countries with a more streamlined and complete healthcare system.
Thank you. So thank you all for your questions and comments. We will move, we're about 8 minutes over now. I'll move to introducing Dr. Mel Whittington from ICER, who is going to present the economic modeling.
Thank you. Hi, everyone. Thanks for being here today. As Dr. Wasfi mentioned, I'll be walking you through the findings from our cost effectiveness work that accompanied this review. One thing to note from the very beginning is since the posting of our evidence report, ublituximab has received FDA approval and a price has been announced. And so the cost effectiveness estimates within this presentation have been updated to reflect this updated information. And then we will carry that information through when we post our final report next month. Next slide. I have no conflicts to disclose. Next. The objective of this component of our review is to estimate the lifetime cost effectiveness of monoclonal antibody treatments for the treatment of relapsing forms of multiple sclerosis. The interventions we included in our cost effectiveness model were ublituximab, natalizumab, ofatumumab, and ocrelizumab. Now, an emphasis will be placed on ublituximab due to its recent FDA approval and it being a new entrant in the class, but we include the other monoclonal antibody treatments where evidence was challenging to differentiate between them clinically. You'll notice rituximab is not listed as an intervention that was included in the cost effectiveness analysis, and that was due to insufficient evidence available at this time on disease progression, which is what the model is driven by. And so we were not able to include that in our economic modeling efforts. Our comparator was dimethyl fumarate. This was selected from numerous conversations with clinical experts stating that it was a market leader, it was effective, and it's currently the lowest cost oral disease modifying therapy. Next slide. We'll briefly discuss the methods before we get into the results. Next. To achieve our objective, we created a Markov model from the US setting. We took two perspectives, the healthcare sector perspective and the base case, and then expanded that perspective to a modified societal perspective in a scenario analysis. And I'll present those results within this presentation. Our time horizon was that of a lifetime with all future costs and outcomes discounted at 3% per year. The cycle length of our model was 1 year, and primary outcomes included cost, quality adjusted life years, equal value life years, and then 2 clinical outcomes that were informed by our conversations with patients and patient advocacy groups. And those were years without ambulatory restrictions, which was defined as time spent in health states less severe than EDSS 5, and then years without a wheelchair, which was defined as time spent in health states less severe than EDSS of 7. Next. Here's our model schematic. You see there's no arrows yet, so just stay tuned for that. You'll see there's 20 health states here. Each oval represents a health state. And so at the top of the schematic, we see EDSS 0 through EDSS 9 for those with relapsing remitting multiple sclerosis. And at the bottom of the schematic, you'll see EDSS 1 through EDSS 9 for those with secondary progressive multiple sclerosis. And then you'll also see a death health state. So the health states were defined by EDSS progression, but the model did also track relapses, which could occur in any of the health states. So relapse was modeled as an event rather than as a separate health state. Next. So now I'll start illustrating some arrows. I do not have all of the arrows on here because there are a lot. So I'm going to first illustrate all of the possible transitions within relapsing remitting multiple sclerosis from one single health state. So you can see there's already a lot of arrows, and this is for one single health state. So these arrows show the health state transitions possible from EDSS of 2. And so you can see from EDSS of 2, transitions can go to less severe health states or to more severe health states or to stay within EDSS of 2. So that same story holds for all of the other health states in relapsing remitting multiple sclerosis. We can go to more severe or to less severe health states. Next. The model also tracks the conversion from relapsing remitting multiple sclerosis to secondary progressive multiple sclerosis. Once in secondary progressive multiple sclerosis, patients cannot convert back to relapsing remitting multiple sclerosis. Next. Now, similarly, here are arrows from one health state for secondary progressive multiple sclerosis. You'll see there are fewer arrows than what was shown for relapsing remitting multiple sclerosis, and that's because these are only going to more severe health states and they can't come back. Next slide. And then finally, from any of the alive health states, the modeled cohort can transition to the death health state, and this could be due to all-cause mortality or disease-specific mortality. Next. Our population included adults ages 18 years and older in the US with relapsing forms of multiple sclerosis. Baseline characteristics are presented on this slide, notably a mean age at 38 and a percent female at 68. You can see at 68%. You can see the distribution among the EDSS states reported on this slide. Next. I want to talk about some key model assumptions that are important to keep in mind when you're interpreting the estimates. Most all of them are related to discontinuation or treatment stopping. So let's take these one by one. First, related to trial reported discontinuation. So for discontinuation for the first 2 years, the trial duration approximated 2 years. So we took the trial reported discontinuation, annualized that, and applied it over the first 2 years. And then the question was, well, what do we do after 2 years? And from conversations with stakeholders and observance of evidence, it did look like discontinuation decreased over time. So after 2 years, after that trial reported discontinuation, we modeled discontinuation related only to serious adverse events and did not vary by treatment. Now we know this assumption is not perfect. We know there's uncertainty in this assumption. And so these estimates were varied widely in the sensitivity analyses we conducted. The second bullet, upon discontinuation, patients transition to a subsequent treatment with cost and effectiveness similar to market-leading monoclonal antibodies. In any model where we might be dealing with treatment switching, it's always a point of contention. As we heard from clinical experts, we heard from patients that if a patient discontinued their initial treatment, they're likely to start another treatment, but that treatment could vary. And then it does vary in the real world. And so we had to make a decision here, and it's important when interpreting this decision, it's important to refer back to our objective and to remember what our objective is. Our objective is not to model the cost effectiveness of a specific treatment sequence, or it's not to recommend the cost effectiveness of a specific treatment sequence. Our objective is to estimate the cost effectiveness of a specific treatment. And so this assumption promotes that objective by being able to isolate that initial treatment that we model. Now, again, we know this assumption is not perfect. We know there's uncertainty, and so we vary the subsequent treatment and scenario analyses that you can find in the report. The last bullet, separate from model discontinuation, the cohort remained on treatment over the lifetime time horizon. We know there's not clear clinical consensus on when patients should stop treatment, but we heard from clinical experts and from patients that they would not want to stop a treatment as long as they were tolerating it. And so we kept on treatment over the lifetime time horizon, but we do include a scenario where treatment stops after an EDSS of 7. Next slide. Here are our treatment efficacy inputs. You'll see in the first column the relative risk of disease progression. These were from the network meta-analysis efforts. So these estimates were applied to disease progression estimates for those who did not receive a DMT. They were assumed the same for relapsing, remitting, and secondary progressive multiple sclerosis. The last column is the rate ratio for relapse rate. These were applied to relapse rates among individuals not receiving a DMT. And again, these were assumed the same for relapsing, remitting, and secondary progressive multiple sclerosis. Importantly, the estimates on this slide are the point estimates. So these are the estimates that were used in our base case analysis. There's uncertainty around all of these estimates. So we have a lot of overlapping confidence intervals when we're looking across treatments, and not all of these point estimates are statistically significant. Dr. Lin already referred to this for ublituximab for that relative rate of disease progression that included 1. So when trying to refer to the uncertainty, I encourage you to look back at the forest plots, particularly for these estimates to see the uncertainty that was included in the model. Next slide. Annual treatment discontinuation, I've already spoke to this a bit, but you can see here in years 1 and 2, discontinuation is higher and varies by treatment. And then in years 3 onward, discontinuation is lower and is held constant across treatments. Next slide. Here are the health state utilities we model that are different for relapsing, remitting, and secondary progressive multiple sclerosis. You see they start out relatively high and then they go down as the disease increases in severity. Additional disutilities were applied for the occurrence of a serious adverse event or for the occurrence of a relapse. Next. This slide shows our health state costs. So in our first column, we see the annual MS direct costs by EDSS state. This includes inpatient care, outpatient care, physician care, durable medical equipment use, and then non-DMT prescription medications. In the last column, we have the annual MS indirect costs. These were used in our modified societal perspective and capture a lot of the non-health economic burden costs that Dr. Lin mentioned. These include a lot of things, including absenteeism, presenteeism, early retirement, premature death, social productivity loss, non-medical costs, paid daily non-medical care, home modifications, special equipment, and healthcare services not covered by an insurance. Importantly, these are for the patient and their primary and secondary caregiver as data allowed. Next slide. These are our treatment costs. So you can see in the first column, we have the wholesale acquisition cost, and then in the middle column, we have the net price per year. Importantly, in the estimates of cost effectiveness I will report, we use the net price per year, so the estimates from that column. Another important thing I want to note about this slide is ublituximab, due to its recent approval, we don't have a good way to know the net price. We have the wholesale acquisition cost now, but we don't have a great way to know the net price. And so for the purposes of our work, we assumed the same discount of ocrelizumab and applied that to ublituximab. So for ocrelizumab, the WAC to net price discount was 23%. We assumed that same 23% discount for ublituximab. So please keep that in mind when you're interpreting our findings, that this is an assumption based on ocrelizumab. We wouldn't think that the discount would be larger than this, but this is an assumption. Additionally, any markup that's associated with provider administered treatments or any costs associated with administration and monitoring are separate from the costs on this slide. Last thing I'll note on this slide is you'll see rituximab is listed here in the second to last row. Rituximab is not modeled as an intervention or a comparator in our analysis, but the estimates are included here for policy relevance. Additionally, we do include a scenario that I will briefly present the results on that includes a hypothetical monoclonal antibody biosimilar. This isn't rituximab, but it does use the annual cost for rituximab in that hypothetical scenario. Next slide. All right, let's transition to the results, and please keep those assumptions and comments I've made in mind when interpreting these estimates. Here are the lifetime model outcomes for our base case. You can see for each intervention as compared to the comparator dimethyl fumarate, there are additional costs, but also additional outcomes in all 5 outcomes that we observed. Next slide. I think it's easier to assess those outcomes looking at incrementally. And so on this slide, it reports the incremental cost effectiveness ratios for each of our 4 modeled interventions. And you'll see across all of these interventions, they exceed commonly used cost effectiveness thresholds, more than double in the best case at the most favorable incremental cost effectiveness ratio. For ublituximab, the cost per quality gained outcome was $451,000 per quality gained, and $403,000 for equal value life year gained. Again, this is assuming a 23% discount off of the announced WAC. Next slide. We conducted numerous sensitivity analyses. From the one-way sensitivity analysis, overwhelmingly the main driver was that hazard ratio on the disease progression for all interventions. And so for ublituximab, given the uncertainty in that estimate, of course, we see a wide range of uncertainty in cost effectiveness. So incremental cost effectiveness ratios range from 130,000 for quality adjusted life year gained, using the most favorable bound of that 95% confidence interval, to being more costly and less effective using the least favorable end of that bound of 95% confidence interval that was greater than 1. Probabilistic sensitivity analyses, the majority of iterations for all interventions were above a threshold of 200,000 per quality or EVLY gained. Looking specifically at ublituximab, 0% were less than 100,000 per EVLY gained, 26% were less than 200,000 per EVLY gained. Next. I'll highlight just a few scenario analyses here. I encourage you to look at the full report. From the modified societal perspective, for all interventions, the incremental cost effectiveness ratios became more favorable by 3 to 7%, still above common cost effectiveness thresholds. Stopping treatment after EDSS of 7, incremental cost effectiveness ratios again became more favorable by 3 to 9%, but were still above common cost effectiveness thresholds. The scenario analysis where we modeled a monoclonal antibody biosimilar comparator. So for this scenario analysis, we assumed an annual price similar to biosimilar rituximab, but because we don't have sufficient data for disease progression, we calculated an average of the clinical data for the other monoclonal antibody treatments and used that as the measures of effect and measures of safety for this hypothetical monoclonal antibody biosimilar comparator. In this estimate, this changed the results dramatically, and it ranged from far exceeding common thresholds, so estimates of around 1 million per quality gained, to being more costly, less effective. And so there's interesting policy relevance around this last comparator, and that this last scenario analysis, and you see when you change the comparator, the story can largely change. And so as we consider additional biosimilar comparators or biosimilars entering the market, this will be an interesting scenario analysis to keep in mind. Next slide. This slide reports our health benefit price benchmarks. Because the clinical evidence was insufficient to differentiate between the monoclonal antibodies on disease progression, and because the cost effectiveness model was driven by disease progression, we report a health benefit price benchmark that is the same across all 4 interventions, rather than a separate health benefit price benchmark per intervention. So the health benefit price benchmark range was $16,500 to $34,900. And so when interpreting those estimates, you can think back to the slide where I showed on treatment costs that included the costs for the monoclonal antibodies, for rituximab, or dimethyl fumarate when you're considering those estimates. Next slide. A few limitations I want to emphasize. The model structure was based on health states defined by EDSS, and this has been critiqued, and that later levels focus too much on physical disability. The natural history studies we use for progression rates for those not on a DMT are more than 10 years old. And so this might make our estimates look more favorable as more recent estimates have slower progression. Variation does exist in the reported quality of life utility scores for people with MS, particularly at high levels of EDS, like EDS 8 and 9. So there is variability there. And last, rituximab was not modeled as an intervention in the cost effectiveness analysis due to insufficient evidence on disease progression. Next. We're grateful for all of the public comments we've received. A few comments to emphasize. One was to use quality of life values less than 0 for EDSS 8 and 9. I've already alluded to how there's a lot of variability between that, but we continue to use quality of life estimates that are greater than 0 for those health states. Comments about stopping treatment once a patient has reached an EDSS of 7, and we have included that as a scenario analysis. And then comments to apply the trial reported discontinuation for each model cycle, so not to model a lower discontinuation over time. And this is something we have varied around a range in our sensitivity analyses. Next. So to conclude, at their estimated net prices, each intervention exceeds standard cost effectiveness levels in the US healthcare system. And the cost effectiveness findings are primarily driven by a treatment's ability to slow disability progression, as well as the annualized net prices. And with that, I think we can open it up for discussion.
Thank you so much, and thank you to you and the whole economic modeling team. Are there questions from the CEPAC? Please, again, use the raise hand function if possible. Steve, Dr. Kogan.
Yeah, thanks. There was a terrific analysis, great response to the questions from public comments as well about the modeling. Thought that was all handled really superbly. Just cut to the chase with my question. It seems like under some assumptions, ublituximab is maybe in the range where we've considered other medications to be fairly cost effective. And not sure that comes through in the report. So would you agree or disagree that perhaps there are, under certain assumptions, a case to be made for this medication being cost effective?
Yeah, I think that's certainly up for interpretation. I think you can look back at our tornado diagram and see that that first row of the tornado diagram does exceed to around 130,000 per quality adjusted life year gain at the most favorable end of the cost effectiveness assumptions. And yeah, so we always encourage not just looking at our base case estimates, but to take that collectively as a whole and to think where clinical effectiveness, where there's evidence available at this time to use as a best estimate in your decision for that.
Other questions from CEPAC? I guess I'm always hesitant to take advantage of my privilege as the chair to ask more questions, but since there's none, can you comment or from anyone else too, on the clinical experts and patient experts about QALYs and equal value of life years in this specific instance? So obviously, I think as most of us know, there's the controversy is, to make sure that, especially when people have disabilities, that we value their years of life equivalently. The argument for QALYs is that you need to capture differences related to health status, not only sort of mortality. In this case, it really does seem from a clinical perspective that health status matters, symptoms matter. So, should we give much to the equal value of life years in this specific instance?
Yes, I think you should always look at the equal value life years gained in every instance. I think, Jason, I think you summarized that really lovely. Now, the reason why the EVLY isn't all that different from the QALY is that the life extension in this clinical area isn't huge. Okay, and so the equal value life year gained measures all life extension equally. But yes, I certainly think the equal value life year gained outcome should be at the forefront of all of your minds.
Other questions from the group?
Jason, do you mind if I just jump in on that and also piggyback on Dr. Kogut's question? Hi, everybody. I'm John Campbell, Senior Vice President for Health Economics here at ICER and a co-author on this work, collaborator with Dr. Whittington. Firstly, so Dr. Whittington presented the health benefit price benchmarks for ublituximab and all the monoclonal antibody treatments that we evaluated. So just know that that's our judgment around this understanding of cost effectiveness. That does importantly include the equal value of life year gained as a measure in that range that Dr. Whittington presented. Importantly, that range was lower than even what our sort of best estimate of a net price of ublituximab currently would be. So Dr. Kogut, I hope that helps you understand where we're coming from with the interpretation of the evidence right now, and also for Dr. Wasfi on our thoughts on the importance of the AVLY as a measure.
That's great, Jonathan. Thank you so much. Rena, Dr. Conti.
Thank you. Mel, I wondered whether you had considered the advent of biosimilar Rituxan in your modeling. And specifically, I noted in the clinical background that actually the majority of prevalent cases of MS are using Rituxan or rituximab. And I also understand that people switch from other therapies onto rituximab or back. So could you tell us a little bit about how you're thinking about the advent of these new therapies into your
I'll take the first pass because this is a really important question, but then Dr. Campbell, Dr. Lynn, please feel free to unmute because this has had numerous conversations throughout our team, and it's a tricky one. So it comes down to we didn't have sufficient evidence on disease progression for rituximab for us to be comfortable modeling that, given that disease progression is the key driver of the model. And so for at least for the modeling purposes, it went back to we don't have evidence. So then I'll refer to the clinical team to round that out.
Yes, thanks, Mel. So yes, that's absolutely right. So the the phase 3 rituximab trial was done in Sweden, and the CDP data, the confirmed stability progression data, was not reliable. We discussed that with actually the trial investigators, and I think Dr. Cohn and Dr. Gould let us know that because EDSS requires trained investigators, and there's a lot of inter-rater variability, and the investigators they had doing that during the trial were not, it was not standardized, and they didn't feel like they actually had good data, that they did not feel it was reliable enough we did not feel that was reliable enough to include as a good estimate of progression. So that's the reason why, you didn't see in our NMA either, you didn't see rituximab appear, because we just didn't feel that data were reliable.
Any other questions or comments?
Yeah, I want to just comment on that. So in the clinical trial, randomized controlled trials, again, many of them are, the majority are recruited outside the United States. Often the investigators are not neurologists, and there is a fee-based EDSS rater certification program. I'm not so sure. I don't think I would say that those Swedish investigators are all MS experts. They know how to administer an EDSS. They may not have all gotten their have recent or updated certification from the very expensive training program out of Basel that you're an investigator in a trial, the company would pay for. But I think the more important context here is that we do have evidence that rituximab slows the disability progression in primary progressive MS patients, right? So the reason all these B-cell therapies exist is because rituximab exists. So Roche and Biogen were co-developing rituximab for multiple sclerosis as a label expansion strategy to be able to keep patent life, to keep that drug alive for their big money maker lymphoma. They had already done it with rheumatoid arthritis, and so it's natural to sort of expand into MS. At the time, the thinking was MS is just RA of the brain. And when they saw those results in relapsing remitting MS, they knew, and I was working for them at the time, that this drug is at least as good as Tysabri. And then came the phase 3 trial data in primary progressive MS that was a near miss. So there was a clinically significant difference in slowing of confirmed disability progression, but it did not hit statistically significance because we were underpowered. At the time, it was very difficult to sort of estimate what progression rates would be in a primary progressive RCT trial population. They took that and I'll just, reiterate, there had never been an MS product that had demonstrated clinically significant difference in disability progression in primary progressive MS before or after, except Ocrevus. They saw that and realized they could make a lot more money if they switched molecules and priced it out at a different price point for the MS market. And that's how ocrelizumab came about. But for us clinicians, we had that MS data in 2007, and ocrelizumab didn't come to market until the end of 2017. So what were we supposed to do in that 10-year period? All we had was natalizumab. And then fingolimod, which we all quickly recognized in clinical experience wasn't quite as effective. So a lot of us started using that medication. So I would just say, in the context of this report, and there have to be rules, and the rules have to be followed, and the clinical disability progression data has to come from relapsing remitting MS populations. And it's true that that data does not exist because the company aborted development of that program and went with a molecule that's almost identical to this molecule and then demonstrated it in that. But we also, in the larger context as clinicians, we know that hitting that endpoint in primary progressive MS is far more difficult than it is in relapsing remitting MS populations.
Thank you. Thank you so much for that. That was very helpful. May I ask a follow-up question, which is, in the real world, will you anticipate that people will switch over to biosimilar rituximab now that it is available?
I think that depends on the insurance carrier. So my understanding in the fee-for-service world is that there's still a lot of insurance carriers that are denying coverage. I think for a lot of patients,
actually, Annetta, I apologize to interrupt. That's a perfect type of question that we're going to be addressing on the policy roundtable when we get into coverage and clinical use patterns. So let's hold that for them.
Got it.
Any additional questions? We have 3 minutes before moving. We're very much on schedule. Greg Lowe. Greg, you're on mute. Or you're not on mute, but we can't hear you.
Apologies. I switched to my phone, so I didn't have the air traffic controller headphones. I'm wondering if we could talk a moment about adherence and what assumptions were made. I've heard insurers who are relatively unconcerned about MS adherence because cynically, I've heard the argument that the cost savings of non-adherence are accruing to the insurer. So they say, why am I going to encourage high adherence rates if it costs me less if they're non-adherent? course, this is never spoken publicly. How's that work with the model, and does adherence, low adherence, is it a wash because it hits the outcomes and the costs equally?
Yeah, great question. I'll first talk about what we do in the model, and then others feel free to unmute and add to the larger conversation. So in the model, we use the treatment effectiveness from the trial. And so adherence isn't modeled directly in there. We're modeling the treatment regimen as labeled, and then the treatment effectiveness as modeled, as reported in the trials, and then from the network meta-analysis. And so we aren't, and then discontinuation as I specified. Does that answer your question, or would more information be helpful? Okay.
Sure, good stuff.
Anyone else? Okay, with that, we are just on time. We are going to move to public comments.